Hi, I'm Giselle and I'm a tutor here at the Ventura College Tutoring Center, and today I'm going to talk about mitosis. So mitosis is a eukaryotic cell division, and what I'm going to go through here first before we get into all these crazy steps is really kind of get a general background information so we can further understand what's going on over here. So I'm going to explain here is the cell cycle. Now the cell cycle is composed of these different things. We have a G1 phase, S phase, G2 phase, and then mitosis. Now I'm going to start right here, for example. And right here, let's think of like a baby cell. You know, a cell just got divided into two cells and now they're babies. And now like any baby or premature organism, it starts developing. And it pretty much just goes through this G1 phase, which is the growth phase of the cell, to pretty much become mature enough to become ready for mitosis. So then it goes down, after its growth phase, it goes through this S phase, which is where DNA synthesis occurs, pretty much where chromatids are replicated to make a sister chromatids. So I'm going to, this is kind of small, so I drew a bigger picture over here. And this is what a chromosome looks like, or what it consists of. So we have, this is one chromatid, and then it replicates itself to make this sister chromatid. Now these two chromatids together are called sister chromatids. This is what generally a chromosome is called, but really a chromosome, it can just consist of one chromatid. The reason we usually are represented as kind of like this X shape is because that's what generally it looks like when it goes through mitosis. But when it separates, they're both identical twins. So these are both chromosomes, or chromosomes really. So that's why I said twins down here, because they're called sister chromatids when in reality they're identical twins of each other. They're both held together by this little structure right here called a centromere. So I'm just giving you kind of like the features of a chromosome so you can further understand when I explain it here in mitosis. After it does this, it goes through the G2 phase, and it pretty much goes into like more growing and development of the cell, and it goes through this phase called interphase. Now in interphase, the DNA develops like a homologous pair, so it starts getting all of its pairs together. It's 23 homologous pairs. Now when I'm saying homologous is because this is a homologous pair right here. So there are two chromosomes right here, and one is from mom and the other is from dad. Okay, so what's going on here, they start pairing up, so when the cell divides, the genetic information is equally distributed within the two cells. Okay, so right here, this is the mitosis phase. So after it goes through interphase, it goes through mitosis. So I'll write it down right here. Really, it will be kind of small, but hopefully you can see it. Mitosis right here, and that's commonly abbreviated as PMAT. Now this is a good way to memorize it. This is the way I memorized it, PMAT, and it pretty much stands for P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. So let's go over here, and I'm going to zoom in and pretty much describe what each of these phases are. So first of all, here it's through prophase, and pretty much the homologous pairs are still coiling up to make really tight chromosomes to become ready for division. The reason why they have to be super tight and very organized is because this is genetic information we're dealing with, and if one piece of DNA gets lost, it could really cause problems within our system. So this is what's going on. As we can see, our nucleus is disintegrating right here. So it's starting to disappear because it's going to need space to really stretch out and like turn into two cells. So then what's going on here is metaphase. Now our chromosomes are tightly compacted, wounded up, and they line up. And what's going on here is these spindle fibers start to attach to the centromere of the chromosome. These spindle fibers provided by the centriole, which we described in our previous video, it gives these chromosomes pretty much the ropes that hold it in, to hold the DNA to pull apart and equally distribute it throughout the cell. Then we go through anaphase, and as we see here, the cell, it starts to lose its round kind of shape, and it's really just stretching out now. And pretty much now these spindle fibers have like teared apart the chromosome, the sister chromatids, to make two individual ones. So I can give you kind of like an example here. Pretty much, let's say like these are, well, I can't really hold it right here. Well, let's say that this is half of it. So we'll say like this is my other half of the chromatid, and here's the other one. Pretty much the spindle fiber attaches to this part, and then like a magnet, it like pulls it out this way. So see how it just distributed into two. So now it's like two separate chromatid, but these are still identical. They're still the same thing. So after anaphase, now we go through telophase, and pretty much it starts really developing, it starts stretching out more. The spindle fibers are pulling as far apart as they can to each side of the new forming cells, and it starts forming this thing called a cleavage furrow. And that's what's going on, is that it starts making a defined line to really get ready to pretty much pinch off and turn into two cells. And that's what cytokinesis is. When these two cells pretty much, they just like boop, pinch off. That's what it is. So then it goes through cytokinesis to, voila, have two new cells with the same genetic information, and they're pretty much ready to go. Before I end this video, I want to make a quick note that in plant cells, this area where they pinch off is a little different. In animal cells, this is what happens. Cytokinesis forms and they pinch off. Now in plant cells, at the end of telophase, which is right here, a cell plate is formed and divided into two cells. So if we can think of it, this is one cell. So say it starts stretching out this cell, kind of going like a rectangular, I guess we could say, and like kind of like an anaphase cell starts stretching out. And then instead of cytokinesis, of pinching off of these cells, pretty much a cell plate is formed in between them, and then we have two new cells right here. So that's what happens. So yeah, this is pretty much what mitosis consists of, and I hope this video helped.